Hi everyone, welcome to our next video for chapter 10. Today we're going to be looking at 10b, which is looking at how we can use structural morphology and molecular homology to look at the relatedness between species. So in our last video, we looked at how we can use the fossil record to determine relatedness between species, and we looked at how we can date fossils to figure out where they sit in the evolutionary timeline. Another way that we can also look at the relationship between species and to figure out what evolved from what and what sits where is by assessing the structural morphology of organisms. So basically what this means is it involves looking for similarities between physical features of species. And we're going to look at the three different types of physical features that we can use to determine relatedness. Before we jump into those, there's two words that you really need to be able to understand. There is homologous and analogous. Homologous means that two organisms share a common ancestor, and the physical similarities that they share is due to a common ancestor. So for instance, in this example here, we've got the wolf, which is the common ancestor. And you can see that these two species here, the fox and the dog, have diverged from the wolf. So they have evolved due to speciation, whatever else, they have evolved from the wolf. And because they've evolved from the wolf, they have similar physical characteristics like the tail, the ears, forelegs, all of those sorts of things. And that's because of a direct ancestor. Analogous, on the other hand, is due to convergent evolution. So this is when there is no common ancestor, but a variety of species have still developed similar characteristics. So for instance, wings. We've got the wings of a bird, the wings of a bat, and the wings of a butterfly. Oh, they should be the other way around. But we know that there is not a common ancestor between these species. So how could this happen? So the whole reason that convergent evolution happens is because of selection pressures. So remember we talked about how selection pressures can lead to natural selection and speciation. Well, there's no common ancestor, but they might have undergone the same selection pressures. So for whatever reason, the bird, the bat, and the butterfly needed to be able to fly, and therefore they all were selected for the phenotype of wings. So they've developed similar features, but they do not share a common ancestor. So it's really important to identify both of these terms. These animals that have undergone divergent evolution are always going to have a more close evolutionary relationship than these organisms here. So, like we said, homologous structures mean that lots of different species share a common ancestor, and even though the function might be very different for these structures, if you look a little bit deeper, you can see that all of these structures are very much the same and have been derived from a common ancestor. And these are, of course, evidence for divergent evolution. So the example is the forearm of a variety of different mammals. So these are all mammals, so we can all assume they have a common ancestor, and these homologous structures provide evidence for that. So you can see that all of the forelimbs for these animals are all very similar structures. So even though they perform really different roles, like the humans use them for grabbing, cats for walking, whales for swimming, bats for flying, you can see that they share the same parts. They've all got this large, this blue structural bone at the start. They've all got the red sort of phalange bits at the end. So the homologous structures, the physical structures are the same, and they indicate a common ancestor, even though the function has become very different due to selection pressures. We then have our analogous structures, which we've said they look really similar and they might behave really similarly and have really similar functions, but they are not derived from a common ancestor. They've arisen due to convergent evolution, which is normally due to similar selection pressures. So the bird and the insect are very, very, very distantly related. They don't share an immediate common ancestor at all, but for whatever reason, they both needed to fly at some point in their evolution. So they both developed wings. But you will see here, the bird wing is very different. It's made up of bones and then these feathery bits, whereas the insect wing has no bones in it. It's made of a much lighter, more flimsy material. So even though they function really similarly, this does not show evidence for a close evolutionary relationship or a common ancestor. We then have vestigial structures. So what these are is they are features that have lost their function due to a change in selection pressures, but we can still use them to determine relatedness between species. So it's basically a feature that we no longer actually use, but the structure of it we can use to determine relatedness. So a classic example here is the human coccyx bone. So we have a coccyx bone in us because we used to have a tail connected at the coccyx bone because of our ancestors, which were the primates and the apes. So we still have this coccyx bone even though we don't use it anymore. And you'll see that that is in the ape family as well. But you'll also see that an example over here, for instance, the snake and the whale, they still have pelvic bones even though they don't walk anymore. So these can also be indicators of a common ancestor at some point in evolutionary history. Even though they've evolved very differently, at some point these two, the snake and the whale, shared a common ancestor that had this pelvic structure. So even though they don't use the pelvis anymore, we can still use this as evidence of relationships. So here's a little test for you. We've got the antennae of a variety of insects. So do you think that this would be an analogous or a homologous structure? It is an homologous structure because even though the antennae look different and they might function different now in the insect species, they all share very similar structural parts. They all have the head capsule, they all have a flagellum at the end, they all have this pedicel and this scape. So even though they function differently and they might look different, they share physical structures that are common and point to a common ancestor as well. We then have the fins of a whale versus the fins of a fish. So this one is, of course, analogous because even though they both share a really common purpose, so they're both used for swimming, we know that whales are mammals and fish are fish, and they're very structurally different inside. So they probably don't share a recent common ancestor. So we've got the whale here, which has got similar to a human forearm in the bone structure. And then we've got a fish fin over here, which you can see is very, very different in the bone structure. Aside from using physical characteristics and structures, we can also use molecular morphology as well. So this is where we analyze the molecular similarities between organisms, whether it be through their amino acid or their DNA sequences. So we only analyze proteins produced from conserved genes because these are the only ones that have remained largely unchanged despite evolution. So we know that during evolution, mutations are going to occur all of the time, but we also know that these mutations may or may not produce a different amino acid because remember we've got our silent mutations, which means that even if the DNA has changed, we can have no amino acid change. Because remember that the genetic code is degenerate, so we can have one amino acid that is coded by many, many codons. So we only look at amino acids in really largely conserved genes, which means there's less mutations. Hemoglobin is a really common example of that, and so is cytochrome C as well. So we can compare these amino acid sequences to determine relatedness. So for instance, in this first example here, we've got the amino acid sequence for hemoglobin from humans, chimpanzees, gorillas, and kangaroos. You can see that the humans and the chimpanzees share the exact same amino acid sequence. You can see that the gorilla has one difference in the amino acid sequence and the kangaroo has several changes. So typically, the more closely related the amino acid sequences are, the more closely related the species are. And you could probably kind of logically tell this. We are obviously human beings a lot more closely related to chimpanzees than we are to kangaroos. So the more differences we have in our amino acid sequences, the more different or the older the common ancestor is between those species. We also can then, of course, look at DNA sequence similarities as well. Would we prefer to do DNA sequence analysis or amino acid sequence analysis? I hope that you can recognize that amino acid sequencing is limited because of those things I talked about. So we share very similar sequences for certain proteins, and we also can't identify those silent mutations that have happened. So the genetic code is redundant or it is degenerate. So if we're wanting to be more accurate and precise, we would prefer to look at DNA sequences and compare them over the amino acid sequences because we're not going to see those silent mutations there. But the principle is the same. The more similar the DNA sequence, the more closely related something is. So you can see between the human, the rat, and the yeast, the rat and the human share more similarities than the rat and the yeast, for example. So we can assume that humans and rats have a more recent common ancestor than the rats and the yeast do. So we can figure out how things are related using structural morphology and also molecular homology as well. So hopefully this helps. If you have any questions, please bring them to class and I will see you there.